Hello guys, welcome to another video. In this video we are going to learn about the micropropagation. What is micropropagation? The technique of producing new plants from single cells, tissue or small pieces of vegetative material. Micropropagation or tissue culture is a technique by which tissue obtained from a plant is cultured in an artificial medium. Micropropagation may be used in herbaceous such as strawberry, gladiolus, tobacco, carnation and gloxina, and woody plants such as apple, rose, kalmia and rhododendron, etc. Then, why do plant tissue culture? A single explant can be multiplied into several thousand plants in less than a year. This allows fast commercial propagation of new cultivars. Taking an explant does not usually destroy the mother plant, so rare and endangered plants can be cloned safely. Once established, a plant tissue culture line can give a continuous supply of young plants throughout the year. For this reason, plant tissue culture is done. Then, terminologies related to plant tissue culture such as callus. What is callus? Callus is a mass of undifferentiated cells that can be induced or arise naturally as a result of wounding. Then explant. Explant is generic term for the living vegetative plant material extracted for tissue culturing on an appropriate medium. Different parts of the plant can be taken as the explant, like entire seed, embryo, shoot tip, a piece of stem, root, leaf, tissue, pollen, grains, etc. Then somaclonal variation. It means heritable variation that arises spontaneously as callus forms on a tissue culture medium. Then clone. Genetically identical assemblage of individuals propagated entirely by vegetative means from a single plant. Then totipotency. Totipotency means the ability of a cell to differentiate and develop into a whole plant when given the correct condition. This is because every cell has the genetic potential of the parent plant. So these are the terminologies related to tissue culture. Then what are the advantages of micropropagation, such as faster multiplication, derived sterile hybrids can be propagated, raised plants pass through an undesirable juvenile phase which is avoided in asexual propagation. Gene banks can be more easily established by clonally propagated plants. From on to many propagules rapidly, multiplication in controlled lab conditions, continuous propagation year round, potential for disease-free propagules. Then inexpensive per plant once established. Then precise crop production scheduling, reduce stock plant space, long-term germplasm storage, then production of difficult to propagate species. So these are the advantages of micropropagation. Then what are the disadvantages of micropropagation, such as specialized equipment or facilities are required, then more technical expertise required, protocols not optimized for all species, plants produced may not fit industry standards, relatively expensive to set up. So these are the disadvantages. Then what is needed for plant tissue culture? Tissue culture, both plant and animal, has several critical requirements such as appropriate tissue. Some tissues culture better than others. Then a suitable growth medium. A suitable growth medium containing energy sources and inorganic salts to supply cell growth needs. This can be liquid or semi-solid. Then aseptic sterile conditions, as microorganisms grow much more quickly than plant and animal tissue and can overrun a culture. Then growth regulators. In plants, both auxins and cytokinins. In animals, this is not as well defined, and the growth substances are provided in serum from the cell types of interest. Frequent subculturing to ensure adequate nutrition and to avoid the build-up waste metabolites. Then steps of micropropagation, such as first step is selection and preparation of mother plant. Sterilization of plant tissue take place. Then next stage is initiation of culture. Explant placed into growth media. Then next step is multiplication. Explant transferred to shoot media. Shoots can be constantly divided. Then next step is rooting. Explant transferred to root media. Then last step is transferred to soil. Explant returned to soil and hardened off. So these are the steps of micropropagation. Here we can see the steps of micropropagation. Then the steps are selection of the plant tissue. Explant from a healthy, vigorous mother plant. This is often the apical bud, but can be other tissue. This tissue must be sterilized to remove microbial contaminants. Then establishment of the explant in a culture medium. The medium sustains the plant cells and encourages cell division. It can be solid or liquid. Each plant species and sometimes the variety within a species has particular medium requirements that must be established by trial and error. Then multiplication. The explant gives rise to a callus, a mass of loosely arranged cells which is manipulated by varying sugar concentrations and the auxin low and cytokinin high ratio to form multiple shoots. Then the callus may be subdivided a number of times. And then root formation. The shoots are transferred to growth medium with relatively higher auxin cytokinin ratios. Then last step is the rooted shoots are potted up and hardened off by gradually decreasing the humidity. This is necessary as many young tissue culture plants have no oxycuticle to prevent water loss. Then these are the steps. The stages of micropropagation. Stages already know that first step is selection. This is the initial step of micropropagation and involves the selection and growth of common plants for about 3 months under controlled conditions. Then next stage is establishment. In this stage, the initiation and establishment of culture is achieved in a suitable medium. Then next step is multiplication. At this stage, the main activity of micropropagation occurs in a defined culture medium. Then last step is rooting. This stage involves the transfer of shoots to a medium for rapid development in a shoots. Then another step is acclimatization. Acclimatization, this stage involves the establishment of seedlings in the soil. This is done by transferring seedlings from stage 3 of the laboratory to the environment of the greenhouse. For some plant species, stage 3 is omitted and non-rooted shoots of stage 2 are planted in pots or in a suitable compost mix. These are the stages of plant tissue culture. Then there are some factors affecting micropropagation, such as genotype of the plant. Selection of the correct genotype of a plant species is necessary to improve micropropagation. Then physiological status of explants. The explants of the most recently produced parts of a plant are more effective than those of the older regions. Then culture media. Conventional plant tissue culture media are suitable for micropropagation during stage 1 and stage 2. However, for stage 3, certain modifications are essential. The addition of growth regulators, auxins and cytokines, alteration in mineral composition is essential. Then light. The photosynthetic pigment in culture tissues absorbs light and therefore influences micropropagations. Temperature. The majority of micropropagation culture necessitates optimum temperature around 25 degrees centigrade. Then there are some applications of micropropagation. First application is artificial seeds. The concept of artificial seeds is being pursued by several biotechnology companies. It entails the mass production of clones of embryos through tissue culture. Then next application is shoot meristem culture. Shoot meristem culture or Mary cloning is another innovative micropropagation technique. It is used to facilitate improvement in certain crops such as orchard crops, which require about 7 years from planting to first blooming. In orchards, it appears that a certain seed fungi association is a normal prerequisite for germination to occur. Then next application is embryo rescue. Sometimes in plant improvement programs, crosses are made between genetically divergent plants. Crossing plants within some species usually has no genetic consequences. The cross is able to develop normally to produce seeds and subsequently plant upon planting the seed. On certain occasions, the breeder would have to import desirable genes from a source different from the species of the plant being improved. Then here we can see the embryo rescue or embryo culture, selection, immature sterilization, sterilized seed, peeling, then growth embryo into a seedling overnight, then plant test after week, ready to transplant in the soil, 2 weeks old seedlings in soil. The whole step of embryo rescue. Then another application is selected crops. Practical applications of tissue culture in horticulture have been widely reported. In garlic, in vitro cultivation has been used to produce quality planting material that is free from viral and bacterial diseases. Then another mass production in vitro virus tree and disease resistant brassica plants occurs. Protoplast fusion, in vitro union of two protoplasts into somatic hybrids. So these are the another applications of micropropagation. And here we can see the artificial seed production. Synthetic or artificial seeds are any totipotent cell, somatic embryos or meristem tips which is artificially encapsulated by chemicals such as hydrogels, which behave like true seeds if grown in soil and can be used as substitutes of true seeds, and possesses the ability to convert into a plant ex vitro or in vitro. Here you can see the artificial seed. Here you can see synthetic seed versus the natural seed. This is natural seed and this is synthetic seed. Then here is the picture of plant tissue culture. Then meristem culture, cultivation of auxiliary apical shoot meristems, particularly of shoot apical meristem, is known as meristem culture. Here we can see the picture of a meristem, lateral bud, apical meristem, leaf, rudimentary leaf, and the application of meristem culture are production of virus-free germplasm and mass production of desirable genotypes. Then orchids. Seeds are exceedingly small with small reserves lose their viability very fast, so they need to be germinated in a nutrient medium. Here we can see different types of orchid which can be propagated by plant tissue cultures. So these are the applications of plant tissue cultures. This is all about plant tissue cultures. No more today. That's all for today. Thanks for watching.